All right, today we're doing something a little different. We're going behind the scenes, stepping into the Andrology Lab, which is the crucial starting point for so many journeys to parenthood. This is where it all begins. So, let's get started. So, think of me as your senior colleague for this tour, okay? You and we're going to walk through this together so you can really understand the massive role this space plays in the science of creating life.
You know, a lot of people think this is just some testing facility, but it's so much more than that. Our job in here, it's to analyze, to select, and to prepare the absolute best sperm for fertilization. It's really a high-stakes preparation ground for the very beginning of life.
All right, so where do we even begin? Well, you might be surprised. Before we even think about touching a microscope, we start with our own eyes. This first crucial step is called macroscopic analysis.
Okay, so what we're looking at here is the seminal plasma. Basically, that's the fluid the sperm are actually swimming in. And the quality of this environment, it tells us a ton about the sample's overall health before we even get near a microscope.
So we're looking for 4 key things here. First up, you see it on the slide, is liquefaction. The sample, it starts as a gel. So has it turned to liquid? Because if not, guess what? The sperm are totally trapped. Then we check the volume, the pH for any red flags, like an infection, and finally, viscosity. Is it super sticky? If it is, it's a real nightmare for us to work with. All right, the first look is done. Now for the main event. We're moving to the microscope for what's called the spermiogram. And here, we're looking at 3 critical things. And we're not just making this up. It's all guided by the World Health Organization's super strict standard.
Using these special slides called counting chambers, we zoom right in on what we call the big 3. The sperm count, their motility, which is just their ability to move, and their morphology, which is their shape. First up, count. And the question here is really simple. How many soldiers are in the army? As you can see, a normal count, that's called normozoospermia. If it's low, it's oligo-oospermia. But the one that's a real gut punch for the patient, that's azoospermia. That means we found 0 sperm. So, having a big army is great, right? But can the soldiers actually move? That's motility. Take a look at this chart. This first group, progressive, these are our superstars. They're moving forward, they've got a purpose. The second group, non-progressive, they kind of just wiggle in place. And the third, immotile, well, they're going nowhere at all. Honestly, the only ones that can get the job done are the progressives. We need swimmers with a mission. And that brings us to the third, and maybe the most shocking metric of all, morphology, which is just a fancy word for shape. Now, of all the millions of sperm in a sample, what percentage do you think need to have a perfect shape for us to call that sample normal? Go on, take a guess. Seriously, got a number in your head? It's just 4%. That's it. Isn't that wild? And the reason that number is so incredibly low is because we judge them by something called the strict Kruger criteria. It is the gold standard, and believe me, it is unbelievably picky. A normal sperm has to be perfect in all 3 parts. You've got the oval head, which is carrying that precious DNA. The neck, that's the powerhouse, packed with energy. And the tail is the motor. Any little defect, especially in the head, and its chances of fertilizing an egg just plummet.
So, the analysis is done. Now what? Well, for treatments like IUI or IVF, we can't just use the raw sample. No way. We have to shift from just looking to actually doing. Our next job is to isolate the absolute best of the best, the champions. This stage is officially called sperm preparation, though, in the lab we usually just call it sperm washing. And the goal here is to get rid of everything we don't want, the seminal fluid, any dead cells, all that debris, and end up with this beautiful, clean, highly concentrated sample of only the most viable, top-tier sperm. So how do we find these champions? Well, we have 2 really brilliant methods. On one hand, you've got the swim up. We literally challenge the best sperm to swim up and out of the sample into a clean layer we put on top. It's true survival of the fittest. On the other hand, there's the gradient method. For this, we use a centrifuge to spin the sample through different layers of liquid, and only the strongest, best-shaped sperm are dense enough to make it all the way to the bottom. So it's kind of cool. We either find the best at the very top or at the very bottom.
Okay, so working in this lab, it takes more than just scientific skill. It demands an absolute, unwavering commitment to a core set of principles. These are our golden rules.
First and foremost, and this is non-negotiable, witnessing. We verify the patient's identity at every single step.
Second, temperature. Sperm are super sensitive to cold shock, so every surface they touch has to be at body temperature, 37 degrees Celsius. Third, no toxicity. That means no perfumes, no lotions, no chemicals. Nothing. And finally, objectivity. We report exactly what we see, not what we're hoping to see. There's just no room for wishful thinking here.
And let me just be crystal clear about that first rule. A mix-up, that is the single worst thing that could possibly happen in this lab. It is a catastrophic, life-altering disaster. And that is why witnessing where a second embryologist has to verify every single step is our most sacred and absolutely unbreakable rule.
Look, if there's one thing you take away from our tour today, let it be this. Think of the Andrology Lab as the kitchen of the entire fertility clinic. We are the chefs, and if we don't analyze and prepare the ingredients perfectly, the final dish can fail. It doesn't matter how brilliant the rest of the team is, absolutely everything starts right here with us.
And that brings our tour to a close. Now you've seen exactly how we analyze a sample and pick out the champions. So I'll leave you with the very question we face every single day. Based on these results, the count, the motility, the morphology, how do we decide which path is right for this patient? IUI or IVF? That, my new colleague, is where the hard data from this lab meets real-world, life-changing decisions.